So friends, till now we have seen general criteria for probiotic selection and different aspects for quality assurance of probiotics. Those aspects include general aspects and functional aspects. The latter part we will discuss in this module. So this module covers health aspects, production aspects and quality control aspect. So let's have a look on first part, that is health aspect. The selection of probiotic organisms depends upon health claims. Probiotic must be able to exert their benefits on the host through the growth or activity in the human body. Most proven probiotic strains are of human origin. A strong case can be made that they are a normal commensals and therefore safe to use. To achieve the health benefits, probiotic bacteria must be viable and available at high concentration, typically 10 raised to power 5 to 10 raised to power 7 CFU per gram of the product. They should provide benefit against gastroenteritis, irritable bowel syndrome and inflammatory bowel disease, diarrhea, cancer, depressed immune function, inadequate lactase digestion, infant allergies, failure to thrive and many more. Now we will explain production aspect of quality assurance of probiotic. It includes acid production, proteolysis, probiotic stability and viability as well as storage. First we will see acid production. The rate of acid development is a critical criteria for the selection of probiotic in milk fermented products. A rapid acid production in the raw material not only helps to prevent the growth of unwanted microorganisms, but is also essential for aroma, texture and flavor of the end product, which has positive influence on its overall acceptability in the form of pH, texture, flavor and aroma of the product. Proteolysis. Proteolysis or casein hydrolysis contributes to the texture and flavor as well as body development in the end product like different varieties of cheese and yogurts. Hence, this criteria is an important consideration for the above products. The ability to produce cell wall bound extracellular proteinases is very important feature of lactic acid bacteria in the hydrolysis of milk protein, that is casein, which supply amino acids to the cell that are essential for the growth of lactic acid bacteria. Protein degradation is mainly associated with Lactobacillus bulgaricus and Streptococcus species. The third criteria is probiotic stability and viability. Probiotic must have the capabilities for its survival in the food, feed and dietary supplements. Manufacturer has given a great attention to probiotic stability. More importantly, the probiotic strain should be stable enough to withstand a conventional industrial production process. Stability is also a strain specific criteria. Probiotic stability is affected by high temperature, oxygen, humidity and high water activity in the culture. The criteria for probiotic selection to study its smooth stability depends on following factors. First, ability of probiotics to survive and maintaining in the storage without loss of viability. Second, ability to grow quickly, maximum concentration in the simple and cheap fermentation medium. Third, ability to grow and survive in microaerophilic or aerobic conditions. Fourth, ability to withstand physical handling without significant loss of viability. Fifth, ability to survive in food matrices and during the processing. Microencapsulation, appropriate selection of acid and bile resistant strains, use of oxygen impermeable containers, two-step fermentation, stress adaptation and incorporation of micronutrients such as peptides and amino acids are the methods used to increase the probiotic stability. Good manufacturing practices, that is GMP, must be applied in the manufacture of the probiotic food and supplements with the quality assurance and shelf life conditions established. The Codex General Principle of Food Hygiene and Guidelines for application of hazards analysis and critical control point should be followed. The last production aspect is a storage. Storage of probiotic supplements at 4 to 5 degrees Celsius is recommended to maintain the viability of microorganisms, and they should be used prior to the expiration date of the product. Probiotic supplements must be kept in the refrigerated conditions, otherwise they have not stay viable. There are some probiotic products which may be shelf stable according to their manufacturers, and their storage and shipping requirements must be met. Friends, last aspect in quality assurance is quality control aspect. The quality control criteria are important in concern with the approval of probiotic over the specific health claims. Thus, functional food regulations should take into account strain properties and their stability during the industrial processing and use. Consideration for the probiotic manufacturing includes quality control procedures such as: first, the criteria and procedures for quality control must be determined and implemented. Second, verification of genetic identity of the selected species. Third, assuring the probiotic potency. Fourth, ensuring the purity of the probiotics, and the fifth one is rectifying the finished product through independent testing. The last part is probiotic labeling. India, for example, is in a nascent stage as far as development and marketing of probiotic product is concerned. Therefore, stringent labeling should be laid down to prevent misleading to the consumers. The term probiotic should only be used on the labels of the products if physiological or health benefits in the human is well established. Regulatory framework established to better address issues related to probiotics including efficacy, safety, labeling, frauds and claims. The International Probiotic Association, that is IPA, and WHO, World Health Organization, provide the guidelines for labeling of probiotics. Under these guidelines, the probiotic product should contain following information like genus, species and strain designation following standard international nomenclatures. The minimum viable number of each probiotic strain should be specified at the level which efficacy is claimed and at the end of the shelf life. Evidence-based health claims should be clearly stated. The suggestions serving size deliver the minimum effective quantity of the probiotic related to the health claims. Nutrition supplement facts, manufacturer lot number and expiry date. Address of the manufacturer, proper storage condition and shelf life should be mentioned. So friends, so far we have studied 5 basic aspects of quality assurance, namely general aspect, functional aspect, health aspect, production and quality control aspect. In general aspect, we have talked about origin, genus, species and strain identification as well as biosafety. Then we have seen various functional aspects like resistance to gastric condition, resistance to bile acid, adherence and colonization of intestinal tissues, antimicrobial activity against potentially pathogenic bacteria and modulation of immune system. Later on, we have started with health aspect and production aspect of quality assurance of probiotics. In production aspect, we have seen production, proteolysis, probiotic stability, viability and storage. The production aspect like stability of probiotics under different manufacturing conditions and food environment is also taken into consideration for their selection. Lastly, quality control aspects have been discussed for desirable characteristic and output of the probiotics. Finally, to summarize this module, learners should note the key points that the selection criteria for lactic acid bacteria to use as probiotic include the following abilities, that is to exert a beneficial effect on the host, it should withstand into the foodstuff at high cell counts and remain viable throughout the shelf life of the product, withstand transit through the gastrointestinal tract, adhere to the intestinal epithelial cell lining and colonize the lumen of the tract. It produced antimicrobial substances towards the pathogen, technologically suitable for industrial process and stabilize the intestinal microflora and be associated with the health benefits. Thank you.